With the sequencing of the human genome now essentially complete, researchers have begun an even more challenging task: the cataloging and analysis of every protein in the human body, an endeavor called proteomics.
What is it that makes life happen? If we could understand the process of life inside living cells, then life science and disease research would be revolutionized, and our ability to understand and treat disease could be transformed. Inside the cells of our body, complex biochemical processes are at work. Billions of proteins and other biomolecules are busily carrying out what is the essence of life. Today, this intricate activity cannot be measured or analyzed. It's hard to tell what these cells are doing. We cannot tell whether they are healthy or diseased. Anima cell metrology is developing a technology to accurately measure this process. We call our technology live proteomics. Here's what it does. Inside the cell, proteins are synthesized according to specific, well-defined plans. Anima's technology will measure and analyze these protein synthesis patterns, revealing the inside story of the living cell. This information is crucial for understanding disease processes and how drugs function. Proteins have an amazing variety of shape, structure, and function, but they're all made in the same way. In each cell in our body, hundreds of thousands of ribosomes, protein production factories of a cell, are constantly producing proteins according to the cell's ever-changing needs. A ribosome produces a protein by chaining together amino acids. The chain then folds into the protein's functional shape. Anima's new technology uses fluorescent tags to label the protein assembly line. With these tags, the ribosome broadcasts light pulses as it assembles the protein. The light pulses form a fingerprint, a unique identifier with specific color and rhythm for each type of protein. This fingerprint is the key to identifying in real time which protein is being produced. Now we understand better what these cells are doing. We have gained an insight into whether they are healthy or diseased. In a sense, we have illuminated this process which lies at the very heart of life science research. Anima's technology will facilitate the work of researchers, therapists, and diagnosticians. It will change the way life is understood and monitored. Anima cell metrology, the next phase in biotechnology.
There is new technological advances that could allow many proteins to be characterized on a mass scale in much less time than it took to uncover the structure of each individual protein in the past.
The code of a cell's gene is read by something called a ribosome, which then helps produce proteins from the genetic code. Some proteins contain receptors that protrude through the cell wall to receive information from incoming molecules. When an outside substance or molecule docks with the protein receptor, the first step in a protein signaling pathway is initiated. This signaling starts a cascade of signals from one protein to the next as each protein folds and changes its shape in response to the signaling. The message ends up reaching the control center of the cell, its nucleus. On another part of the cell, another molecule docks with a new protein receptor. This signaling pathway begins, but it is blocked by another element in the cell. Scientists are working on developing specific drugs that can unblock signaling pathways. Once the pathway is repaired, the signaling can continue on to the cell's nucleus. This would be a successful use of a drug in the new field called clinical proteomics.
While there may be as many as 1 million different proteins, most are just variations on a handful of themes. The same shared structural moffat, barrels, helices, molecular zippers are found in the proteins of plants, insects, and humans.
Proteins are polymers constructed from the same set of 20 different amino acids. The primary structure is the specific sequence of amino acids specified by the gene. In the secondary structure, portions of the protein begin to coil and fold into unique three-dimensional formations. Two kinds of secondary structure units are the alpha helix and the beta-pleated sheet. The folds and coils of the protein are the result of hydrogen bonds that occur at regular intervals. The tertiary structure is formed by interactions between side chains of various amino acids. At this stage, some proteins are complete. Other proteins incorporate multiple polypeptide subunits, creating what is known as the quaternary structure.
Proteomics is a new approach that will enable analysis of proteins and comparisons at the protein level.